Hello everyone, welcome back to Practically's NEET Bio class, and today we're going to learn about proteins. Okay, so now we are going around in this chapter, we are going around a lot of biomolecules. Okay, what are biomolecules? Biomolecules are something which actually those compounds, those molecules which are help, which make our lives, okay, which we are made up of, proteins, carbohydrates, lipids, etc. Okay, we are all made up of these kinds. Our cell has all kinds of these molecules. Okay, so these are the molecules which give us life basically. Okay, so these are the biomolecules. Now, in the previous class, we were talking a little bit about the micromolecules, the smaller size ones. Okay, so these micromolecules are something which make a macromolecule. Okay, I think you'll remember. The thing is, small micromolecules together, they make a macromolecule. So we were talking about amino acids. Now, amino acids, when there are around 20 essential amino acids and there are other non-essential amino acids and all, together, when they come together, they make proteins in different shapes, different sizes, different ways. Okay, they like turn each other, okay, turn on each other. Somehow they make a ball, like a very serious kind of structure, 3D structure out of it, and out of that a protein gets formed. Okay, so today we're going to discuss about protein. Now let's see who has joined. Hi Himmat, hello, good evening. Hi Anish, hello. Thank you everyone for joining. So let's get started on proteins. Now, before that, let me talk a little bit about ourselves. We are from Practically, and it's an app, okay, and it is a wonderful app. You can learn everything, okay, all the science-based subjects, science, maths, all kinds of simulations, videos, everything. You don't have to like go into other Google-based information. You'll find everything over here, okay. And you can also like, share, and subscribe to our YouTube channel, okay, where you can get, if you subscribe, you'll get to know what and all is happening in the channel. Okay, you'll get to know for NEET, we have bio, we have chemistry, we have JE maths also, everything is running around. Okay, so do check it out. You can also like us on our Facebook and our Instagram page. Okay, so proteins. Now, the protein thing, okay, the first time somebody actually talked about protein was this person called Berzelius. Okay, so Berzelius was the one who first talked about protein in the year 1838, and it was used by J.G. Mulder the first time in 1840. So what are proteins basically? Now, whatever we eat, a lot of it, protein is something which gives us the strength. Okay, it gives us strength. Protein is something which gives us strength. When we want to build our body or something, you're going to the gym, you want to build your body, always you'll see nutritionists, dietitian people, they always talk about how you should have a lot of protein. Instead of going for something carbohydrate based, you should go for something protein. Why? Because it will give you strength. It will give you muscle mass. Okay, everything else will just like you'll gain weight, but not a lot of muscle mass and all. Protein is something which will give you proper muscle mass, proper strength and everything. That's why it's so important. It is such a, and the one of the most important macromolecule. Macro, not micro, macromolecule. Why? Because it is heavier. Okay, it is larger in size. Okay, now 15% of the protoplasm is actually made up of protein. 15%. We have a cell, there's a membrane. Okay, now they have protoplasm and everything, and there's nucleus. Okay, there's cytoplasm. Now, whatever it is inside the cell, whatever it is, that is the protoplasm, and 15% of it, okay, 15% of the protoplasm is actually proteinaceous in nature. So you think how much protein we have. Now there are proteins, we know how amino acid look. Okay, now amino acid will have a carbon, we will have hydrogen, will have an amino group. Okay, we'll have a carboxylic acid and an R group. Okay, this can change, this is variable. Now we know that it has some kind of nitrogen, see, amino group, it has carbon, okay, it has oxygen also, it has hydrogen also, it has sulfur also. Okay, so a lot of things are actually present in small quantities in a protein. Okay, so there is nitrogen, of course, carbon, of course, oxygen, hydrogen, CHO will always be there. Okay, carbon, hydrogen, oxygen, even nitrogen in case of protein, everything will be there. Apart from that, there will be some kinds of minerals like sulfur, iron, phosphorus, copper, calcium. These are also there in very minor quantities. Okay, so this is how a protein is. Okay, how it looks, what it is made up of, and all. Now, proteins are basically polypeptides. Okay, now what are peptides? How do we know what are peptides? Why are we talking about peptides? What is a peptide bond? Okay, now if you ask that, we know that proteins are made up of amino acids. Okay, now amino acids, just now I showed, looks like this. Okay, H, amino, carboxylic acid, and an R group. This is variable. Okay, now what happens is this amino acid, okay, one amino acid, when it attaches to another amino acid, this carboxylic group will attach to another amino acid. Okay, so the carboxylic group of one amino acid and the amino group of the other amino acid, there is a link. Okay, this link which attaches two peptides or two amino acids is known as peptide bond. Okay, so what it is? One amino acid plus one amino acid, this bond, okay, is the peptide bond. Okay, now each protein is a polymer of amino acids. Okay, together there will be different amino acids coming together. Okay, somewhere it will be cysteine, 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 cysteine. Somewhere it will be alanine, cysteine, proline, tryptophan. The different kind of amino acids together, or same kind of amino acids together. These are known as the proteins. So when there are same kind of amino acids joined together, they'll be homopolymer. Okay, so what is homopolymer? Same amino acids together forming protein. Okay, heteropolymer, different amino acids. Okay, so now amino acids can be essential also, can be non-essential also. The thing is, our body does not make essential amino acid. Okay, our body does not make essential amino acid. What it does, we take our amino acids from our diet, food, from plants, animals, etc. Okay, so because our body cannot make essential ones which we actually need, our body cannot make. So this comes from the food. Okay, and there are different things proteins can do. Okay, proteins are such, just now I said, such strength they give. They do a lot of functions in the organisms. Okay, there are nutrients across cell membrane, they fight infections, there are hormones, there are enzymes, everything in all of these things, protein is present. Okay, protein is present. So see how important proteins are. Everything. Apart from, like, protein does not work in genetics. Okay, they don't carry genetic material. Okay, apart from that, everything, starting from starting from helping in digestion of food to transporting things, okay, transport of minerals and transport of hormones and transport of other molecules, everything, in everything protein is there. A lot of life functions they do. Now, collagen is the most abundant protein in the animal world, and rubisco, ribulose biphosphate carboxylase oxygenase, is the most abundant protein in all world. So the most abundant protein in the world is rubisco. Okay, let's see. Hi Shravan. Okay, hi Neha. Okay, Himmat, my name is Shabnam. Okay, so if you have any doubts, please let me know. Now, the structure of protein. This is one of the most, like, a lot of questions you get in NEET, okay, regarding the structure of proteins. It is one of the most important topics when it comes to proteins in this chapter, biomolecule chapter. The structure of protein is very important. Okay, so what happens? So we know proteins are made up of amino acids. Okay, so what happened just now? I said, so what happens? The amino acid, the carboxyl group of one amino acid, see, this is a carboxyl group of one amino acid, okay, and there is the amine group of the other amino acid is there. So when the peptide bond has to be, like, created, this bond has to be created, what happens? Together they leave a water molecule out. One H from here and one OH from here. They leave out and they make a peptide bond. This bond is known as peptide bond. So together a lot of, suppose, amino acid A, amino acid 1 peptide bonds with amino acid 2 has bonded with amino acid 3. This is how a protein is made. Together 10, 20 they'll put, okay, 10, 20 amino acids they'll put and they'll form a protein. Amino acids will attach with the help of peptide bonds, one after the other, and they'll form a protein. Okay, so peptide may be dipeptide, the bonds may be dipeptide, tripeptide, polypeptide, okay, any number of peptide bonds might be there. One of the simplest protein is insulin. According to Sanger, insulin consists of 51 amino acids, and proteins can have up to 4 level of conformation. Like, they'll only have 50. So what happens with insulin is, one second, yeah, insulin, what happens? It is very simple. The structure is very simple. One amino acid is linked with other amino acid with the help of peptide bond. So like this, 50 will be there. Now what they do, they twist and turn each other. There'll be twists and turns within each other, okay, and they'll have a 4-level conformation, a 4-level, okay, conformation. So it is a simple one only, but the structure it looks is a bit complicated because it has twisted and turned itself. Okay. So now the structure of protein can be divided into 4 levels. Okay. So there is primary structure, secondary structure, okay, tertiary protein structure, and quaternary. Okay, so one of the most simplest formation structure in protein is the primary structure. It's very easy to remember. What happens is there'll be amino acids. Okay, suppose these are amino acids. They'll be linked with each other with the help of peptide bonds. Suppose these are peptide bonds. Okay, just one after the other, one after the other, one after the other, and peptide bonds. That's all. Okay, so a linear sequence. This is a linear sequence. Okay, one after the other with the peptide bonds they will be. And some things like ribonuclease, just now I said insulin has 50 peptide bonds like that. Hemoglobin, these are all simple primary structures. Now the sequence of amino acids, okay, which is the first amino acid, which is the second amino acid, and so on, is called the primary structure. So what happens, suppose there is a tryptophan, leucine, serine, cysteine, and phenylalanine, something like this. This is some kind of a protein. Okay, so 1, 2, 3, 4, 5. This is some kind of a protein. Now, these things only, if they do, okay, 3, 2, 1, 5, 4. Just you change the places of the amino acids, this will be a different protein. Because its position is different, the amino acids have changed their position, so this will be a different protein. Okay, so a protein is imagined as a line. The left end is represented by the first amino acid and the right end is represented by the last amino acid. Okay, so the left one is the first amino acid, your right one is the last amino acid. Okay, so there will be a N terminal here, okay, and a C terminal over here. It will always have a terminal, no, an amino group, a carboxylic group. In the ends, there will be like that. You have to see where the N terminal is, that is the first amino acid. Where the carboxyl terminal is, that is the last amino acid. Okay, and accordingly, if you change the positions of the different amino acids, your protein will keep on changing. Okay, same amino acids only. If you just change the position, your proteins will start changing. Simple idea. It's not very complicated also. Different amino acids held with peptide bonds. Okay, that is the primary structure. Now, suppose this is 1, 2, 3, 4, 5 amino acids you have. You change the structure 3, 4, 1, 2, 6 like that, and the protein will change. Okay, so that is how it is. Now, secondary structure, as I said, primary, secondary, tertiary, quaternary. So the secondary structure is a bit more complicated. Okay, it's not as simple as the primary structure. So what happens in the secondary structure? Okay, it is folded, local folded structure that form within a polypeptide due to interaction between the atoms of the backbone. Okay, so suppose there are a lot of amino acids. Okay, they have peptide bonds. Okay, but in between these only, they'll have different amino acids are linked to with some kind of other peptide bonds also. They linked with different far away amino acids. Okay, so they are linked to their closer amino acids also. Also, they are linked to the far away amino acids by some peptide bond. That is the secondary structure. Okay, so the backbone just refers to the polypeptide chain apart from the R groups. The R groups remain different. The two most important structures are alpha helix and beta pleated sheets. Okay, and both of them are held in a shape by hydrogen bonds which form between the carbonyl O of one amino acid and amino H of another. Okay, so they are held by the hydrogen bonds of each other, of different amino acids. Okay, so how it looks, see here this is alpha helix. So we know helix structure, we know how DNA looks like, right? Somewhat like this, it will look somewhat like this. So what will happen? Similarly, in proteins also, it will be like that. So what happens? The carbonyl of one amino acid is hydrogen bonded to the amino of an amino acid that is 4 down the chain. Okay, so see, in this picture you can see there are lots of peptide bonds. Just the R thing is outside. The R portion, the different portions, the variable portion is outside. The rest of the groups are inside. Now suppose this is position 1. Okay, this is one amino acid here. So see, there is the amino group. Okay, position 1 amino group is hydrogen bonded with the position 5 carboxylic group. This is position 1, this is 2, this is 3, this is 4, this is 5. Okay, so they are like literally related among themselves to the far away carboxylic acid also. Okay, so that is how the the alpha helices, they make helices, because they're related to their near ones also, they're also related to the far ones also. Okay, so this pattern of bonding pulls the polypeptide chain into a helix structure, okay, like a curled ribbon, and each turn contains 3.6 amino acid. So here you will have around 3.6 amino acids. Each helix, this will have around 3.6 amino acids. Okay, so that where that push we have between amino acid in position 1 with the carboxylic acid in position 5, you have that bond which brings the amino acids together and they form a helix structure. Okay, because they're so attracted with that hydrogen bond, they make it into a helix structure. And as I said, the R group, that is the variable group, will always be out. Okay, so the variable group, the variable amino acid will be always outside. All the R's are outside the helix structure. Okay. Hi Sai, welcome to the class. Okay, so today we are learning about proteins. Now, so beta pleated sheet, that was alpha helix. Now beta pleated sheet. Now, just imagine you have proteins on paper. Okay, in paper like structure, you have proteins. So one paper on top of another paper, suppose, and see, this is a protein, okay, made up of all peptide bonds and amino acids and everything. You have another paper, same thing. Okay, so there are sheets, and these sheets are then linked by hydrogen bonds. Okay, so these papers, okay, one after the other, they are linked by hydrogen bonds. That is how a beta pleated sheet looks. One paper beside each other, on top of each other, but they are linked by the hydrogen bond. Different sheets of paper linked by the hydrogen bond. That is how a beta pleated sheet looks. Okay, two or more segments of a polypeptide chain line up next to each other, forming a sheet-like structure, a paper-like structure, okay, held together by hydrogen bonds. The hydrogen bonds form between, of course, the bond will always be, it will never be amino-amino, or it will never be carboxylic carboxyl. Okay, it will always be amino with a carboxyl. Okay, no amino will never bond. Amino carboxyl will never bond. Okay, it will always be amino carboxyl. Okay, and the R groups extend above and below the plane sheet. R group will always be outside, be it beta pleated sheet, be it alpha helices, it will always be outside. Okay, now the strands of beta pleated sheet may be parallel. Okay, if they are one on top of each other, they'll be parallel and pointing in the same direction, or anti-parallel, pointing in the opposite direction. Okay, they might be one on top of each other or by side. Okay, they might be parallel or they might be anti-parallel. How the sheets are placed, it depends on that, and accordingly the protein will also form. Okay, now that was secondary. Now, so tertiary structure. Tertiary structure is nothing. Okay, just imagine primary structure, and then what happens? As I told you, the R group are always outside, something like that. Now you bond the R groups together. Okay, till now we were bonding the amino carboxyl. Here, there will be amino carboxyl, will be there. But with that, we'll have the R groups also bonding with each other. Okay, and then that kind of structure forms a tertiary structure. So the overall three-dimensional structure of a polypeptide is called tertiary structure. The tertiary structure is primarily due to the interactions between the R group. Okay, so this is not one, there will always be peptide bonds, but this is peptide bonds. Peptide bonds are there plus interactions in the R group. Okay, so the R group interactions that contribute to the tertiary structure, including hydrogen bonding. So what kind of bonds are formed between? It depends. It totally depends what that R group is. Okay, so there is hydrogen bonding, ionic bonding, dipole-dipole interaction, London dispersion forces, different bonds are formed. Okay, out of that, one of the most strongest bond which is formed in this tertiary structure is the disulfide bond. There is sulfur bonding. That is one of the most strongest bond, and it is seen in cysteines. Okay, in cysteine structure, you can find sulfide bonds. Okay, and this is one of the most strongest bonds ever. Okay, so this is how the tertiary structure. Now finally, quaternary structure. This is also something, this is all three-dimensional look and all. Okay, this is also something, it is simple only, but it is made to look a bit complicated. What happens? Many proteins are made up of single polypeptide chain and have only 3 levels of structure. So one after the other, polypeptide chains are made. Then there are, on that polypeptide chain, you make some alpha helices and beta pleated sheets and everything. However, some proteins are also made up of multiple polypeptide chains known as subunits. And these subunits come together, they form the quaternary structure. So what happens? Suppose there is one alpha helices subunit and one beta pleated subunit. Okay, another alpha helices and another beta pleat. So together they'll form a protein. They'll all form, these are subunits, but they'll form, subunits will come together and they'll form the protein. Okay, such as hemoglobin. Okay, it carries oxygen in the blood and is made up of 4 subunits, 2 of alpha helices and 2 of beta pleated sheets. There are others such as DNA polymerase. They are also composed of 10 different subunits of proteins. There will be proteins only, but these simpler proteins will come together and form a bigger structure. That is the quaternary structure. Now, see, this is how it looks. Suppose this is the primary structure, one after the other, one after the other, one after the other. This is a peptide bond. Okay, now they together, they'll fold. Okay, they'll make some bonds between the first and the fifth position, fifth and the 10th position, something like that. And they'll form bonds among the farther amino acids and they'll form the alpha helices. Similarly, they'll like make sheets out of each other and then bond with the different kinds of sheets, that is beta pleated sheets. Okay, now these actually will then twist and turn. Okay, they will twist and turn among each other and they'll form the tertiary structure. How will they twist and turn? With the help of the bonds between R groups. Okay, with the help of the R groups forming disulfide linkage, hydrogen bond, ionic bond, they'll twist each other. Now, all of these twisted subunits of protein will come together and form a bigger structure, that is the quaternary. Small subunits coming together. Okay, I hope you understood. If you have any doubts, you can leave it in the chat box. Hi Yuvraj, good evening. Okay, so now proteins are, that was the structure of different proteins. Now proteins are again classified according to different like shape and like what they are attached to, how simple they are, how complicated they are. Accordingly, they are classified. So on the basis of shape, there is a fibrous protein. See here, this is fibrous protein. Okay, so these are insoluble in water. Animal protein are resistant to proteolytic enzyme and is spirally coiled, such as hair protein, keratin, you know, right? So hair protein or keratin looks like this. Okay, now this is like straight and a little bit twisted, something like that. Then there are globular proteins, which are a bit round or spherical in structure. This is more of an oval or spherical in structure. Those are known as globular proteins. These are soluble in water. Polypeptides coiled about themselves to form oval or spherical molecules, such as albumin. Albumin is present in eggs. Okay, insulin, hormones like ACTH, oxytocin, etc. So it is roundish. This is more of a straight wavy kind. Okay, straight wavy kind, and this is more of a roundish kind of protein. This is on the basis of shape of the protein. Now further on the basis of constituents. Okay, now suppose only simple proteins, as in they are only made up of amino acids. Okay, the there is a stretch. It can be a bit complicated also in the shape, or it can be straight, but it is made. There are no extra substances. There are no other molecules. Okay, so there are no other molecules here. It will only be amino acids. Now there are conjugated proteins. They are complex proteins combined with a non-amino acid substance, such as a prosthetic group. Okay. And then there are the derived proteins, which are hydrolyzed by acids, alkalis, enzymes, the products. Okay, these are not in real, like they are proteinaceous only, but they are derived from a mother. Okay, derived from a mother protein. These are all processed. Okay, and conjugated protein, these have an extra non-proteinaceous substance added to it. Okay, so this is okay. Yuvraj, can you tell me what you want me to explain first? Which one do you want me to explain? I'll explain accordingly. Okay, just put it in the chat box what you want me to explain, exactly what you want me to explain, and then I will help you out. Hello, Netra. Good evening. Now, we said that according to what is added into the protein, okay, how it is added into the protein, like what is there, an extra substance might be there. Okay, apart from the amino acids, some extra substances might be there in the protein. Okay, so accordingly, they are also divided. Suppose there are some nucleic acids, some genetic material, okay, some nucleic acids, etc., found in chromosomes, ribosomes, etc. With the protein substances, there are some nucleic acids also. Then it's a nucleoprotein. Suppose there is a large amount of carbohydrate. There is more than 4%, a large amount of carbohydrate are there, such as mucin. They are known as mucoprotein. Okay. But then when there is a small amount of carbohydrate with the protein, that is known as glycoprotein. That will only be around 2 to 3%. Now, suppose there are some pigments, some colored, okay, some kind of extra color pigments there is, such as hemoglobin. Hemoglobin has what? Has that red color. Okay, so those are chromoproteins. Lipoproteins, very easy. They have lipids. The name is only suggesting they have lipids in the protein structure. There are some lipids also. So it is a lipoprotein. Metalloprotein, what? There are some metal compounds attached to the protein. Okay, such as how like iron, zinc, copper, etc. they have in the proteins. Okay, so that is known as metalloproteins. Hi Yuvraj, can you just tell me what doubt you had? I will try to help you out. Okay, so now that was what extra part it was there, what extra molecule might be there. Okay, that was conjugated protein. Now, basis of nature of the molecules also you can divide the protein. It can be some some acidic also. It can be acidic in nature, such as an anion. Okay, they exist as anion and include acidic amino acids, such as blood groups. They are acidic proteins. Okay, now then there can be basic proteins also. They exist as cations, and they are rich in basic amino acids. Basic amino acids as in whose pH is a bit higher, like on the after 7, 8, 9, 10, etc. Those are basic. Okay, so such as lysine, arginine, these are all basic amino acids. So the proteins made up of different kind of basic amino acids is known as basic protein. Okay, so this is how. Okay, if HCl is not in our stomach, then what will we have? See, HCl is an acid which helps in the digestion. Okay, digestion and process of the food particles, the bolus one. When we eat, we have a lot of things. We have proteins, we have carbohydrates, we have lipids and everything. Suppose there's no HCl, then it won't, the process will not be there. Okay, the process will not happen. It will be deficient. Your food will not get completely digested. Okay, so that might be a problem. Okay, I hope, although it is not related to this chapter, maybe I helped you a bit. Maybe we'll discuss about it when we discuss more about the digestion in humans or digestion in animals and all in the next chapters. Okay, maybe we'll discuss about it a bit more. Okay, now last, what are the functions of proteins? Now, as I said, the proteins are made up of a number of things. Okay, proteins are like different, they have amino group, they have this thing, a carboxylic group. Okay, they are acidic, sometimes they are basic. Okay, they have an R group. Okay, which can be variable, which can be any kind of group. Okay, so they have, because of this very, like, a very unique kind of structure, they can do a lot of stuff. Okay, a lot of functions they have in our body. Okay, such as what? Proteins occur as food reserves, such as glutenin, globulin, casein in milk. We can store food. Okay, and they are the most diverse molecule on earth. That is because of their unique structure. Okay, because of their unique structure, they are very diverse in nature. Okay, because of that R group only, whatever is R, it can keep changing, right? It can change. So because of that change, it can be very unique. Okay, protein work as hormone, as insulin, glucagon, etc. These hormones are proteinaceous in nature. Even enzymes. Okay, enzymes are also, the enzymes which we have in our body, they are also proteinaceous in nature. Okay, they are structural component of the cell, because it a majority of the protoplasm, okay, protoplasm is made of protein. Okay, a majority of the cell, whatever protoplasm is, a majority of it is made by protein. Okay, they are biological buffers. Okay, what they act as catalysts, such as enzymes, etc. They help in reactions. Okay, reactions and stuff, life reactions, life processes. They are kind of a biological buffer which helps in certain reactions in our body. They are also helping in defense. Okay, they actually, they can neutralize pathogens. They neutralize pathogens. Okay, we have some foreign particles such as bacteria or some virus and all. There are some proteins in our body which actually neutralize their effect, which actually stop their effect. Okay, that kind of work is also done by proteins. Okay, so now if you have any doubt, please put it in the box. Okay, and we can also go back and then, okay, we can put some ideas, and we can, if you have any doubts or something, you can put it in the chat box and we can also solve some questions. Okay, so which of the following proteins was sequenced by Frederick Sanger? Just now we read Frederick Sanger. He sequenced insulin. Okay, he was the one who said insulin is a simple structure made up of around 50 amino acids. Okay. Next, what is a bond between amino acids called? Amino acids is called what? A peptide bond. Okay. So, R, H, carboxylic group, amino group, this bond, okay, between amino and the carboxylic group is known as the peptide bond. This is the peptide bond. Okay, so that is the peptide bond. Next, which of the following statements is true about proteins? Proteins are polymers of glucose? No. Why will proteins are polymers of amino acids? Okay, proteins are polymers of amino acids. Proteins are polymers of peptide bonds? No, because peptide bonds are just bonds. Okay, proteins are polymers of disulfide bridges? No, this is also wrong. Okay, so the proteins are polymers of amino acids. Next, which of the following is not the function of proteins? It helps in digesting food, of course. It carries the genetic information? No, that is the work of nucleic acids. Okay, fights against invading pathogens? Yes. Okay, the antibodies and all, they are all protein based. That helps in invading pathogens, help in transporting oxygen in blood. Of course, they do that. The proteins are help in translocation. Okay, so it does not carry genetic information. Okay, thank you everyone for joining today. I hope you liked the class, and please come back every alternative days. We have classes for bio, for NEET. Okay, class right now we are doing class 11. After we're done with class 11, we jump on to class 12. Okay, so now like every Monday, Wednesday, and Friday, 7 p.m. we have this class. Even on Tuesdays, Thursdays, Saturday, we have the chemistry class for NEET. Okay, don't forget to like, share, and subscribe to our channel. Okay, and I'll see you guys later. Bye bye and have a good night.